IB Biology, we're now looking at the functioning of enzymes. We'll be taking a look in this video at metabolic pathways, energetics, and activation energy. A PDF of this presentation is in the description down below, along with a quiz for you to test your understanding of the topic. Enzyme molecules are proteins. They have very large globular shapes, shape being an important aspect of any discussion on protein molecules. They're often conjugated and indeed with transition metals. Therefore, we find copper, zinc, manganese there, but also we might find magnesium. The primary function of an enzyme is to reduce the activation energy of a biochemical reaction. And because the activation energy is reduced, this means that there's no need for extreme pHs or extreme temperatures that you might otherwise require to get a chemical reaction to occur, such as we might do in a chemistry laboratory. Therefore, life, because of enzymes, can function in moderate conditions and at speed. An enzyme is, as a protein, is produced in response to the expression of a gene. And this will turn the substrate molecule into what we call the product molecule. Substrate is what a chemist would call a reactant. When this occurs in biological pathways, large chemical changes, large radical chemical changes are not common. Instead, what we tend to see are small changes which add up to cause large changes. An example of which is seen in linear metabolic pathways. Here, A is the initial substrate, is turned into product B by enzyme E1. B would be turned into C by a separate enzyme, C into D, and then D finally into the desirable product, the final product, by another enzyme. So lots of small changes adding up to the final product which is desired, an example of which is seen in the process of glycolysis in respiration. Alternatively, we will come across cyclic metabolic pathways, an example of which would be the Krebs cycle, another would be the Calvin cycle. Here, a molecule enters the cyclic changes to be accepted by molecule B. They will combine under enzyme action to form molecule C. In turn, C will be transformed into D, and a product E will be formed in the next step. Characteristically of a metabolic pathway, there will also be regeneration of the acceptor molecule. These ideas are explored in more detail in the Calvin cycle and in the Krebs cycle. If we take a look at the energetics of these reactions, often when substrate is turned into product, heat is liberated. From the perspective of mammals and birds, this is involved in maintaining body temperature, so we become homeothermic, and we regulate this heat production, and this has profound consequences for the efficiency of their metabolic pathways, but also at an ecological scale for their distribution around the globe. Metabolic pathways are a combination of anabolic and catabolic. Anabolic means to build molecules, and this usually requires the input of energy. And that such a reaction is known in biology as being endergonic. So free energy is required to be put into the reaction. And this is illustrated in this diagram here. Catabolic reactions, contrastingly, break down molecules. Energy is often released, and we refer to this as exergonic, the release of free energy, energy that can carry out work or change. Either of these two pathways, exergonic or endergonic, has the following characteristics in terms of the energy of activation. The substrate sits here with an energy. So the substrate molecule is sitting at this energy level, but to catalyze the change to the product here, a large activation energy hill is necessary to be overcome. In a chemical reaction within the laboratory, you could deploy pH, acidic conditions, strong alkali conditions, or high temperatures. But within a biological cellular situation, instead this is achieved by the presence of the enzyme, which reduces the activation energy, allowing the reaction to proceed fairly quickly and without the necessity for extreme conditions. So life on Earth is, the chemistry of life on Earth is occurring within fairly moderate conditions. That said, there's always an exception which tests the rule in biology, and we do find the existence of what are known as extremophiles, illustrated here with this picture of a geyser in Yellowstone National Park, where the temperatures in this particular location here are in excess of 80 degrees centigrade. But that's for a different part of our study. Links to additional videos in this sequence are appearing now in front of you, along with subscription and links back to the Play channel for you to look at other videos.